So this is my talk about finding evidence from population studies as to whether wildlife is actually being harmed by chemicals to the extent that we might suppose from ecotoxicology information. And soon I hope some of the thoughts that are in this talk will come out in a new ET&C focus paper. So if we were to extrapolate from our ecotox data results into what we believe might be happening in the wild, we often get very concerned. We've had the talk from Peter and his group about organic chemicals in European waters. There's also concerns, as you know, about endocrine disruption and wastewater effluent, and this has an effect on NGOs and then politicians. However, our job of scientists is to look for actual evidence of harm in the wild. So if we were to look for historic trends in field data, what would we actually see? Well, it's not so easy to find the data we might be looking for, despite the importance one might attach to it. This is an example of some data recorded by an angling club in the lower part of the River Thames in the UK, and it shows for this fish, the bream, that the weights of fish that they've caught on average have steadily risen from 1950s to 1977. So things seem to be improving. Here we've got much more recent data from rivers in Germany, collected partly with the help of the German Environment Agency, also looking at bream, and the trends are showing an increase in weight of the fish and in fact in condition factor improving from the 1990s till today, despite all the chemicals we actually release into our rivers from sewage effluent. This is data from several British rivers, and it's actually trying to normalize angling data. So this is catch per unit effort for anglers in the UK against time. And you can see that there's a general upward trend. In other words, there's more fish apparently being caught per angler in the 2000 period compared to 1975. So another example of apparently improving numbers of This is a different set of data. It's not fish, it's macroinvertebrates. It's a slightly complicated figure from a paper by Avon and Ormerod. And what it's showing is that over time, from 1990 to 2008, the diversity or species richness of macroinvertebrates have improved across the range of British habitats, but most particularly in urban habitats, again where you might have expected the most exposure to sewage effluents to be occurring. So it appears to be another example of positive change for wildlife. However, perhaps if we look in a bit more detail at sites where we expect the highest exposure to sewage effluent to occur, we might then see population level effects. Now we can start this off thanks to modelling, which we can do to predict the exposure of any reach in a river to sewage effluent. In this case, this is estrogens from sewage effluent in the River Thames. So regulatory agencies such as the Environment Agency in the UK have monitoring programs for counting the number of fish in rivers, and they do this in many rivers in the UK each year in the summer. And what we're trying to do is link this to fish population data. So being scientists, we start with a hypothesis, and we're asking, is the average fish abundance for any fish species will be negatively correlated with sewage effluent or estrogen exposure. Here you can see some data for a common fish in British rivers, the roach. We're looking at 41 separate river sites from the Rivers Aire, Calder, Avon, Great Ouse and Thames in the UK, and it's the average number of fish over 10 to 13 years, and we're comparing it with the exposure at those locations to sewage effluent or estrogens. And you can see from this figure that there's no relationship between the two whatsoever. So we falsified the first hypothesis. Sewage effluent content in British rivers does not depress average fish populations. So you've seen it's not correct for roach. It's not also, we don't see any relationship for bleak, dace or perch. So just refining our examination a little bit more, if we look at whether populations are harmed in years with much higher sewage effluent exposure, such as might occur in particularly dry summers, we can look at temporal trends. Before we do that, it's important to know that the fish that are counted in our rivers, this is an example of the age structure or demographics of fish in a typical British river, is dominated by very young fish of 1 year, 2 years plus. So what we're going to try and do is to compare the conditions of the preceding 12 months to when the fish were sampled. This is an example of changes in numbers of roach in the River Thames at Caversham. So this is the top graph on the left. You can see we start off in around about 2000 to 2013, and the green line is showing fluctuations in the roach population. The red line is telling you what the average estrogen or effluent exposure of the preceding 12 months was. So if we plot those two bits of data as an XY scatter graph, we actually see, rather surprisingly, a positive relationship between the roach and sewage effluent. So we get more roach when we have years of apparently more effluent exposure. So our second hypothesis seems to be false. Where we've looked in detail at the Middle Thames and Great Ouse, the roach populations reacted positively, in fact, to higher sewage effluent exposure, not negatively. So if we now think about how do macroinvertebrate families change over time with respect to sewage effluent, so we're trying to do something similar as you just saw with the fish. This is one site on the Thames, and we're comparing the black line, which is the effluent or estrogens, against fluctuations in different families and invertebrates. In fact, we can't find positive or negative relationships with these invertebrate families and effluent exposure at any of our 9 sites. So the conclusions, it would appear that the chemicals in domestic effluent in the UK from 2000 onwards are not harming our common lowland fish populations, nor are they having overwhelming effects on invertebrates. It's important to note I'm using the word populations because it may be that individuals may be being harmed, but the populations are still being sustained. We do see reduced invertebrate diversity associated with effluent exposure, but invertebrate families do not respond to temporal changes in effluent exposure in a consistent way. So a reliance on laboratory ecotox tests may be misleading us, and that wildlife populations are much more tougher and adaptable in the face of chemicals than we thought. So I like looking at wildlife populations because they integrate the effect of multiple endpoints and mixtures of chemicals. Now some say that we can't interpret population studies because it's all too complicated. And admittedly there are many factors that influence a population in the wild. But no one used this argument when the dog whelk mollusk was being wiped out due to TBT, or the Asian vulture was being wiped out thanks to diclofenac. So my view is that monitoring populations of aquatic wildlife over time is the single most important activity in our field of environmental science. Thank you.